Congratulations on making it through the first lecture exam. I'm really proud of you guys and your hard work in easing into physiology and getting through digestive physiology. We're going to be shifting gears and we're going to be thinking over the next few weeks about cardiovascular physiology. And I wanted to start this out by reviewing, sorry, my dog just let out a huge sigh. I'm really sorry about that. We were going to start reviewing the anatomy of the cardiovascular system. We'll break that word cardiovascular apart and think about cardio, the heart, and vascular, those blood vessels. So I wanted to kind of give you an overview of what we're going to be doing and get into the learning objectives for this particular lecture. So just to kind of look at what's coming up, so this is week 6. I'm posting this lecture about blood vessels and the heart. I know it's a little bit late in the week. I'm sorry about that. I had some issues with recording, but the slides have been posted, so hopefully you've gotten a chance to look through them a little bit. So this is the online lecture. For one of my classes, I know you guys didn't have class on Monday, and the other class you are taking your lecture exam finally. So we're just kind of easing into this unit with an anatomy review. During week 7, the in-person lecture will be on EKG and the cardiac cycle. The online lecture will be on hemodynamics, so the flow and physiology of blood, kind of as a more holistic connective tissue. Then over the next few weeks, we're going to be talking more about the lymphatic system, different types of blood cells, the function of blood cells. So as we get into week 8, we'll start thinking about the actual material that's flowing and the physiology of different specific types of blood. So the lymphatic system and blood specifically. When we're talking about hemodynamics, we're thinking about the flow of blood and blood pressure through a bigger organism. One thing that I wanted to clarify is that this online lecture should be viewed before we do the in-person or online lecture in week 7, but both the in-person and online lecture for week 7 can be viewed in any order. I know sometimes it makes more sense to do the in-person lecture first and then the online lecture, but for week 7, they are different enough that you don't need to worry about doing them in any particular order. That being said, during week 8, you should watch the online lecture before the in-person lecture. Okay, so as we're going through this lecture, I encourage you to think back to anatomy and try to identify gaps in your knowledge about the cardiovascular system to really help build up that foundation for cardiac physiology and hemodynamics. You should be very comfortable with the names and locations of the atria and ventricles, the basic chambers of the heart. You should know the different types of valves. So there's the left and right atrioventricular valve, the aortic valve, and the pulmonary valve. The aortic valve and the pulmonary valve especially are going to be really important to know when we're talking about hemodynamics. The left and right atrioventricular valves are going to come into play quite a bit when we talk about the cardiac cycle and EKG. So there's a lot of different synonyms that we use for those. I've tried to include them so that you kind of have an idea of what we're talking about. We'll also get into the structural and functional distinction between arteries and veins. There are a lot of misconceptions about arteries and veins, whether they're oxygenated or deoxygenated, the histology and the flow. So we'll really try to get into that and clarify any confusions you might have, and then get into different types of arteries and different types of capillaries. Those different types of arteries are again going to be very important when we talk about the cardiac cycle and hemodynamics. I really wanted to spend a moment focusing on different levels of capillary leakiness, because when we get into filtration and talk about homeostasis later in the semester, having that foundation in those structures is going to be very important. And finally, I want you to become very familiar with the cardiac conduction system. You should be able to map the flow, the electrochemical flow of a signal from the SA node, which we'll get back to at the end of the lecture, all the way to ventricular contraction. And being able to do that and overlay that with the anatomy of the heart should really set you up for the EKG lecture. I know I've said the term EKG a couple of times. I'll clarify what that means at the end of this lecture. To start out with a brief review of cardiovascular anatomy, here I'm drawing material from chapters 19 and 20, so kind of hopping around. If you want to read those, especially the chapters that are like anatomy of blood vessels and anatomy of the heart, that might be supportive in your understanding of this material, but as always, you're only going to be tested on what is in the slides and my lectures. So remember, one of the goals I have for you in this class is to really practice the language of biology, not just memorizing terms, but really taking the time to break apart words. So when we look at this term cardiovascular, we can break it apart into cardio and vascular. Cardio is specifically referring to the heart. If you think about cardio, like the exercise that gets your heart pumping. A cardiac surgeon is a surgeon for the heart. So you should know that that prefix cardio is telling you something to do with the heart. And we'll get back to vascular in a moment. So we're going to focus first on the anatomy of the heart. When we're thinking about the anatomical structure of the heart, there's a few different things to look out for, and I'm going to kind of show you this anatomy right now, have you look at that image on the right side of the screen, and remind yourself kind of first of all about anatomical positioning. Remember that whenever we look at diagrams in anatomy and physiology, it's like we're looking at a patient sitting in front of us or lying on an operating table in front of us. So our right is going to be their left and vice versa. That means that the right atrium is positioned in the top left of your screen, or on the side of the diagram. So you should remember those atria are the top chambers. There's the right atrium and the left atrium, and material just kind of flows into these. There's not necessarily going to be valves there that control flow the way that there are for other heart structures. So we have blood flowing into the atria, and then we have blood being pumped out of the ventricles. So this is kind of tricky because we have these vessels, the aorta and the pulmonary artery, that are connected to these ventricles, these chambers that are on the bottom of the heart, basically. But they have these vessels, again, the aorta and the pulmonary artery, kind of grouping towards the top. So they're actually connecting with chambers in the more inferior portion of the heart, but these vessels all kind of group towards the superior portion of the heart. So I know that that can get kind of tricky if you're not familiar with that anatomical structure. Remember that it's always a good idea to orient yourself to the image by remembering that because the ventricles have to pump so hard, their myocardium, their heart muscle, is a lot thicker. So you can actually see this in the bottom of your screen over here. So we can see that that muscle is a lot thicker on the ventricles. So just really reminding you about how everything connects together. We'll talk about different valves and kind of different flow over the next part of this lecture. So when we're thinking about those valves, there's valves that exist between different chambers and also valves that exist between ventricles specifically and blood vessels. When we're thinking about atrioventricular valves, that word is a mouthful, but we break it up into atrio for atrium, those top chambers of the heart, and ventricular for ventricles, the bottom chambers of the heart. So atrioventricular valves are valves that separate out and control the flow of blood from the atria to the ventricles. On the right side of the body, there's the right atrioventricular valve, which is also called the tricuspid valve. It's called the tricuspid valve because if you look over here, you can see that there's these three parts to it, whereas the bicuspid valve on the left side of the heart just has two parts to it. So tricuspid on the right side, bicuspid on the left side. So we have the right atrioventricular valve, which is called the tricuspid valve, the left atrioventricular valve, which is called the bicuspid valve, and it's also actually called the mitral valve. So if you hear mitral, bicuspid, left atrioventricular, those are all referring to the same the same valve. There's also valves that exist. They are shown surrounded by this orange box on the right side of the screen that control the flow of blood from ventricles to vessels. So there's the aortic valve, which separates out the left ventricle and the aorta. The pulmonary valve separates out the right atrium, or not right atrium, sorry, the right ventricle and the pulmonary artery. So arteries are connecting to ventricles because ventricles are pumping blood out of the heart and arteries move blood away from the heart. So those main vessels that connect to the heart are the aorta, which connects to the left ventricle, and the pulmonary artery, technically the pulmonary trunk, that connects to the pulmonary valve. Remember that pulmonary means lung. So to just briefly kind of connect the structure of the heart and the valves, when they're closed, we can kind of see closed here. Open looks like this. So we see those heartstrings, those chordae tendineae. We see kind of how they change and how they influence the movement of the heart valves. So there's actually different types of cells within cardiac muscle, and so that differentiation is important for the movement of the heart, so this function of beating, of pumping, and the type of electrochemical impulses that control and initiate that movement. So as we're thinking about the cardiac cycle and we're thinking about this, contraction and relaxation, it's important to remember that cardiac muscle is super unique in that it has this property of autorhythmicity. It can not only, conduct these electrochemical impulses, it can also initiate them. So it's important to know about different types of cardiac cells. There's the myocardial contractile cells. Myocardial should tell you myo for muscle, cardial for heart, so cardiac muscle or heart, the heart muscle. Contractile means that these are the ones that are involved in actually contraction cells. So these are the cells that are involved in contraction of that cardiac muscle. There's also myocardial conducting cells, which tell you that they're probably doing a little bit more of this conduction system, so more of the action potentials, that depolarization and initiation and conduction of signals. So most of the cells in our myocardium, in our heart, are in the atria and the ventricles, are these myocardial contractile cells. So by just the number of cells, 99% of them are myocardial contractile cells. So these can conduct impulses, they can kind of receive these impulses. Their purpose is to kind of receive those impulses and contract heart muscle. So as we talk about the cardiac conduction system later in the lecture, and especially in the EKG lecture, you're going to see kind of how this conduction serves to initiate that contraction. So that remaining 1% of cells in the atria and ventricles are those myocardial conducting cells, and those form the conducting system. The next slide kind of shows you a visual of some of those, the Purkinje fibers, but these tend to be pretty small generally with some protein filaments. Purkinje fibers tend to be pretty big, so there's kind of like a variety of types of cells here. And these are responsible for initiating and propagating that action potential. So they are key to this idea of autorhythmicity. But to help visualize that, oftentimes when we're looking at an image or a diagram of heart muscle, of heart tissue, we're looking at those contractile cells. But if we zoom in on the histology and we kind of identify certain structures, we can see especially the Purkinje fibers because those tend to be pretty large. So if you see Purkinje fibers labeled on a histological slide, you should know that those are involved in the cardiac conduction system. Cardiac muscle is really unique. It has a lot of properties that seem pretty similar to skeletal muscle. It's striated, it has these kind of stronger contractions, but it's involuntary. You can't think about your heart necessarily and just control its beating. You can change other factors like maybe taking deeper breaths or, other things that you kind of intuitively know adjust your heart rate. We'll get back to that in just a moment. But in kind of understanding how the heart functions, if you look at a histological slide of cardiac muscle, you can see these intercalated discs between different cells. So those intercalated discs are super important because in order for an electrochemical signal to be sent, there has to be some electrochemistry there. And by that we mean ion flow. And so this heart muscle has to have these openings, those gap junctions, to allow ions to flow through and continue that signal. That idea is going to make a little bit more sense when we spend more time going through nervous system physiology. But we did briefly talk about this in the first lab when we talked about why we call that electrochemical, this idea of sodium ions and potassium ions. And you might have learned about this in depth in Bio 5 or anatomy, but I don't want to assume that you kind of know exactly how an action potential works. But not only do we have this flow that's important between different cells, those cells still have to be strongly attached together because remember the goal of this tissue is contraction and relaxation, which is very dynamic. And I should have included a GIF of a beating heart here. I'll show that to you in another lecture. But if you look at videos of beating hearts, it is such a dynamic process. It is very violent and visceral, and that's why we have so many extra protective layers and pericardium and different fluids to protect our heart from that movement and friction. Otherwise it would, tear itself out of us. So those cells have to remain attached together, and desmosomes do that. Those desmosomes are a special type of connection between different cells within tissue that allow kind of flexible but strong movement. So they keep them anchored together but allow the muscle to kind of move. So you have this coordination of signal through the gap junctions and then a coordinated movement using the desmosomes. So as that happens, when you have a relaxed heart, you have much larger volume, so you have it maybe filling with blood. And then when it contracts, that reduces the size of those chambers, the atria and the ventricles, depending on whether you're talking about atrial contraction or ventricular contraction. When we're thinking about getting blood to most of our body, we're thinking about ventricular contraction. So when you have that contraction, you're squeezing the blood out of the heart, shooting it into the vessels. It's a little bit more complicated than that. We'll talk about the cardiac slide or the cardiac cycle next week. But this idea of relaxation and contraction is summarized by the words systole and diastole. Systole refers to that period of contraction. When we're talking about ventricular contraction, we're talking about blood being pumped into circulation in the body. When we're talking about atrial contraction, it's getting that blood into the ventricles. Diastole is that period of relaxation that's occurring as chambers are filling with blood. So being able to fully contract and then fully relax is really important in the cardiac cycle in order to make sure that we're being efficient with our blood flow. So we'll talk a lot about that again next week, but you should start considering that there's a process of systole and diastole for both the atria and the ventricles. Those separate chambers are relaxing, they're filling with blood, and they're contracting, they're pumping blood out into circulation kind of separately from one another. So as we start to look at the depolarization events that kind of trigger that contraction, we start to see these different patterns that emerge. So one of our goals next week is going to be to ground that electrochemical pattern with the cardiac cycle. That's what this image on the right side of your screen is depicting. Those little P, Q, R, S, and T wavy lines are characteristic parts of an EKG. We have the P wave, the QRS complex, and the T wave. There's going to be different parts of atrial depolarization and repolarization, as well as ventricular depolarization and repolarization at different points in that little diagram. So again, we'll talk more about how that diagram is generated and how it relates to the cardiac cycle next week. So when we're thinking about how we go about actually quantifying our heart's work, there's a formula that you're going to come across, and as you're studying for the T's or maybe studying for other classes, you're going to see this formula CO equals HR times SB. So that CO stands for cardiac output. It's kind of how we measure the work that our heart is doing. Heart rate, HR, is how frequently our heart is pumping. And stroke volume is a very particular volume that we'll talk about when we talk about the cardiac cycle and hemodynamics, but this is how efficiently our heart is pumping, basically. So I think we've all had the experience of having our heart rate be pumping, or like be very, very fast. Maybe we get nervous, maybe someone cuts us off in traffic, or we turn around and our kid is gone somewhere at the store and we don't know where they went. Or maybe you are working out and you're really doing a lot of cardiac activity, so your heart starts pumping. So there's a lot of situations, or like maybe you're just sitting down for a long time, you stand up too quickly and all of a sudden your heart is pumping, all kinds of situations where our heart rate can change. So that is very flexible. It's affected by neurotransmitters, the nervous system, our mental state, our age, our fitness, different types of medication. So in general, people who do a lot of cardiac activity or cardio activity, people who run a lot or swim a lot, tend to have much lower resting heart rates. And part of the reason for that is that their heart muscle is a little bit more efficient. They have a higher stroke volume. So that is kind of grounded in our physiology. It is determined in part by our biological sex, our fitness, our heart size, and kind of how much we make efficient and work out that cardiac muscle. So people who run marathons all the time have very efficient stroke volumes, so they have much higher stroke volumes. They can have a correspondingly lower heart rate and still get the same cardiac output, because cardiac output is a product of heart rate and stroke volume. Conversely, if you have a very low stroke volume, you have to have a much higher heart rate to achieve the same cardiac output. So to kind of think about how that blood gets out of the heart and then to the rest of our body, we're thinking about that cardio part of cardiovascular as well as the vascular part, which is referring to the vessels. So we'll start thinking about different types of vessels, starting with kind of generally different types of vessels, focusing then on arteries and then on capillaries. So remember that in general, we characterize arteries as moving blood away from the heart. So arteries away, veins return blood back to the heart. Remember that there is a pulmonary circuit and a systemic circuit. Oftentimes we focus more on the systemic circuit, but I wanted to really remind you about some stuff in the pulmonary circuit throughout this lecture because it pushes back on a lot of the assumptions people make about arteries and veins. So functionally, arteries serve to move blood away from the heart, veins return blood back to the heart. Because they're under very different stressors and they accomplish very different things, they also have a lot of structural differences as well. In general, the lumen, or the, I was going to say cross-sectional lumen, the cross-sectional diameter of the lumen of the arteries and veins tend to be a lot bigger coming out of the heart, and then it gets smaller and smaller. So we have a higher surface area to volume ratio as we transition from arteries to arterioles to capillaries. That higher surface area to volume ratio means we have a lot more surface for exchange between the blood, those vessels, and the surrounding tissue. As that transition happens and blood continues to flow and is then draining and returning back to the heart, we start getting into venules, and then the diameter gets bigger and bigger as we get back into veins. So when we're thinking about the histology of these different vessels of arteries and veins, we have on the right side of your screen, there's a diagram showing, it might make more sense if I kind of annotate this a little bit. This outer portion is kind of like the outer portion of the artery. So there's tons of cells visible here. Each of these little guys is a cell. So this is showing tissue layers. So just so you have an idea of the size and scale. And then on the inside here is where you would expect to find blood. So that's the lumen, that's the inside of the artery. This area right here is the arterial wall. And then the vein is the same kind of setup. We have the outside where the body tissue would be, and then the inside, the lumen, where the blood would be. So we have those different layers of tissue, and they're labeled very clearly in that top diagram. And then in the bottom diagram, we have an actual histological cross-section. So one thing that stands out about this histological cross-section is that the overall diameter of the artery is smaller, but it tends to stay much more upright. It maintains that circular shape, that round shape, in these histological slides, regardless of the overall diameter. So we have a much thicker wall. It's much more reinforced. It has more muscle relative to the vein. So even though this vein has a wider diameter, it tends to look kind of floppier because it doesn't have that muscular reinforcement. So one thing that I wanted to point out is that I have a lot of students who say that arteries are reinforced because they're carrying oxygenated blood. And that's true for the systemic circuit, but that is definitely not true for the pulmonary circuit. So when we look at the pulmonary artery, which is displayed, it's labeled as PA and then has an orange circle around it on the right side of your screen. When we look at the histology of the pulmonary artery, that is carrying deoxygenated blood, but it's still just as reinforced as a systemic artery. So part of the reason for that is, remember, when we have the pulmonary circuit, we have the right ventricle pumping and sending blood through the pulmonary trunk, then the pulmonary arteries to our lungs. That blood is deoxygenated, but it's still under a good amount of pressure because it's coming from a ventricle that is actively pumping. Now, the right ventricle does have slightly less muscle than the left ventricle, but it's still under a large amount of pressure. It has to keep that continuous flow going, so that pulmonary artery has to be reinforced. So regardless of whether you have oxygenated blood or deoxygenated blood, this idea of pumping and then having that reinforcement and that continuous flow is crucial for arteries. And the same is true for veins. The systemic veins are deoxygenated, they're floppy. The pulmonary veins are oxygenated and also floppy. So clearly the fact that it's oxygen doesn't have anything to do with the structure of the overall vessel. It's really its connection with the heart. The veins are moving blood back to the heart. They're not under pressure. They're relying on movement from skeletal muscle and on those valves to prevent backflow to get the blood back to the heart. And it's just going to kind of drain into the atria. So in this situation, again, whether it's deoxygenated or oxygenated, doesn't make a difference. The veins are under a lot less pressure. They're floppy, whether they're carrying deoxygenated or oxygenated blood. So now that we've kind of distinguished between the structure and function of arteries and veins, within arteries, we distinguish between elastic arteries and muscular arteries and then arterioles. So I'll kind of walk you through each of these. I do want to state though that this transition is not like as clear as first elastic, then muscular. It's a slower, gradual transition in tissue and histology as we go from elastic to muscular. So these are kind of both structural and functional distinctions, but it's not as clear as individual sections of the artery. So these elastic arteries tend to be found closer to the heart. They tend to be much higher volume in their lumen, so higher cross-sectional diameter. And because they tend to be more elastic, they're able to kind of expand and deal with pressure and kind of like accommodate that higher volume and maintain a pressure gradient as blood is entering from the ventricles. We contrast that with the muscular arteries, which are farther from the heart, have a lower volume in their lumen, and are less likely to be able to maintain a pressure gradient. They tend to be a little bit more rigid. So as we have blood coming in, if those were closer to the heart, you wouldn't have like a nice pressure gradient moving blood along. It would disrupt flow. So these play a very important role farther away from the heart, where you have more vessels, so higher surface area, but a lower volume in each vessel. This is really important for maintaining vasoconstriction at these lower pressures. And then finally, we have arterioles. Arterioles are an important transition point between arteries and capillaries. When we talk about hemodynamics, we're going to discuss resistance and that idea, and blood flow and blood pressure, kind of the relationship between all of those. Arterioles are very important for that. They also kind of tend to be consistently contracted to some extent. So there's a consistent muscular or vascular tone. So if you hear that term vascular tone, it's referring to the tone or kind of muscle properties of the muscle in those tissue layers of the vessels, in this case, the arterioles. So when we're thinking about kind of the flow between arteries, specifically between arterioles and capillaries, it's not as simple as everything just flowing through into the capillaries. It's very carefully controlled. So for example, if you have tissue that is dead, that is no longer functioning, it's not going to be very helpful for you to send blood to that tissue. So you might control the flow of blood using sphincters that kind of maybe if they are contracted, prevent blood from flowing from the arteriole to the capillary. If they're relaxed, then maybe blood is going to flow through. So this idea is very important for hemodynamics. It's important for ideas about resistance and blood pressure. And then these sphincters are also going to be important when we talk about filtration, calculating glomerular filtration rate, and understanding that interplay. So also along those lines of filtration, it's very important to take a moment and pause and understand capillaries. When we talk about, again, homeostasis and balance, edema, filtration, leakage, understanding the leakiness of capillaries is going to be very important for that process. So we have this endothelial layer that is lining the capillary itself. And then surrounding that, we have that basement membrane. So if we have kind of a if we have less leakiness, if we have maybe a continuous capillary or even a fenestrated capillary, there's not going to be as much leakage or filtration out of material from that capillary. But if we have a sinusoid capillary where we do have incomplete basement membranes, where the gaps in the endothelial layer line up with the gaps in the basement membrane, then we have material leaking out of the capillary. We call that a leaky capillary. Those are, again, very, very important when we think about glomerular filtration. So I want you to keep that in your mind, keep that in mind when we go on to the excretion system. So I wanted to kind of briefly remind you of flow in the systemic circuit and the pulmonary circuit. I'm going to take a moment to say that I was planning to kind of diagram this process on the next slide, but my pen, my stylus is not working as well as I had hoped, so I'm not sure how that's going to work out. I will also post a video to kind of help you, remind you of that flow in case you're struggling. But the systemic circuit, the goal of that is to move blood that is oxygenated away from the heart to body tissues, and then after there's exchange, to have deoxygenated blood from those tissues moving back to the heart. So that's the systemic circuit. It goes from the left ventricle through the aorta to the systemic arteries, and then systemic arterioles, and then capillaries, transitioning back to the systemic venules, the systemic veins, draining in through the vena cava and entering the right atrium. The pulmonary circuit moves that deoxygenated blood from the right ventricle, it's getting pumped out through the pulmonary trunk, which breaks into the left and right pulmonary arteries. That's going to go to the different lungs for reoxygenation. So it's pulmonary. That word should be familiar to you as lungs. So pulmonary is going to move that deoxygenated blood from the right ventricle, small pump, high pressure, but not as high as the aorta, going to the lungs where oxygenation is going to take place, and then that blood is going to be brought back from the lung to the heart through the pulmonary veins. So here I just wanted to very briefly introduce this idea of a kite diagram. You might have learned this in anatomy or previous classes, and I'm sure from anatomy the anatomical view of the heart is very familiar to you. But just in case you're still struggling with how those different chambers connect with different vessels, I wanted to teach you this diagram. You can skip ahead if it's not going to be helpful for you. But basically we start by drawing a kite. That's a very ugly kite, I'm so sorry. But this is going to represent the heart. So we're going to divide it into 4 chambers. Remember that this is the right side, this is the left side. So we have the right atrium and the left atrium, because the atria are at the top of the heart. Then we have the right ventricle and the left ventricle. Down here, we're going to have that systemic circuit, and we have the pulmonary circuit. So this diagram is kind of helpful for, again, remembering how those different chambers connect with different vessels, especially since all the vessels kind of get tangled at that superior portion of the heart. So it gets a little bit confusing what's attached where. But we have the left ventricle attached to the aorta. And again, that's sending oxygenated blood to the body, to body tissues, and it's going to be moving through those systemic arteries until it makes its way to the capillaries. So there's really high surface area. We have oxygen going into body tissue for cellular respiration, and carbon dioxide, a waste product of cellular respiration, is going into the capillaries. Remember that it's not just respiratory gases that's being exchanged here, it's a lot of other material, but when we're focusing on oxygenated versus deoxygenated blood, which is a big part of what we're going to be doing in respiratory physiology, we're thinking about relative partial pressures of oxygen and carbon dioxide. So now we have this deoxygenated blood that's going to make its way back up from those capillaries through systemic venules and then into veins. And it's going to make its way to the vena cava, which are going to drain into the right atrium. Remember that veins always connect to the atria at the end, ultimately, and then ventricles connect to arteries. So we have these veins returning blood back to the heart. They're going to enter into the right atrium, and remember that's the vena cava. There's the superior vena cava and the inferior vena cava, so it's a little bit more complex than this. But then we have blood flowing through that right atrium, through the right atrioventricular valve into the right ventricle, and then it's going to be pumped and sent out through the pulmonary trunk. Which again branches off into the right and left pulmonary arteries. Those are going to make their way up to the lungs, which I didn't really save enough room for this. That's going to be your right lung and your left lung. Just trying my best over here. So we're going to have that blood going through this little loop-de-loop to the lungs. Remember that the lungs have a ton of surface area as well. So there's going to be all those different capillaries wrapped around the alveoli. So there's going to be a ton of gas exchange. This time we're going to have oxygen going into the blood. And we're going to have CO2 leaving the blood. And we're going to be breathing it out. So remember, that's not a perfect process. That's why you can still give mouth to someone and give them enough oxygen to kind of help them breathe, because you still have a good amount of oxygen in the air that you breathe out. It's just that the relative partial pressure of carbon dioxide is higher. So now this blood is nice and oxygen rich again. It's going to flow through the pulmonary vein into the left atrium. So then it's going to flow through the left atrioventricular valve and then be pumped out again through the aorta. So that, I hope, integrates those chambers with the vessels, kind of the loop-de-loop that happens. I know that sometimes when you focus on the anatomical structure of the heart, you get really bogged down in the details, and it's easy to forget how this all flows together. So I hope kind of listening to that and watching it get built up is a little bit helpful for you. I know that my graphic design here is not great, but I will again post plenty of Khan Academy and Crash Course videos and probably Amoeba Sisters, so that you get a little bit better, higher caliber visuals for how this process is happening. Okay, so now we're going to get into the cardiac conduction system, and this is really where the crux of cardiac physiology starts. So understanding how those cardiac conducting cells, the Purkinje fibers, different things like that, come together to make a unique structure that's able to initiate and propagate those action potentials in the heart is key to understanding the cardiac cycle. So I'm going to start out by showing you that cardiac conducting system, some of the structures that are involved, and then we're going to start to kind of visualize it in more video format. So that cardiac conduction system is again responsible for initiating those electrochemical signals that eventually result in contraction of the heart. So we have the sinoatrial node, which let me kind of overlay this with an actual anatomical image of the heart so you can visualize that a little bit better. So towards the right atrium, we have the sinoatrial node. And then between the two atria towards the ventricles, we have the atrioventricular node. And we have the atrioventricular bundle, the atrioventricular bundle branches, Purkinje fibers, and that's the system. So I probably should have paused there. Hopefully you can pause the video and take that into consideration. So we have this general flow. You should know the order of the cardiac conducting system, so kind of how electrochemical signals are sent. I'm going to show you a GIF that does a good job of showing the actual signal, and then I'll show you a GIF that kind of ties that together with the cardiac cycle, how contraction happens, and how different chambers are filling and pumping out blood at different rates according to the cardiac conducting system. So here we have the different parts of this labeled. We have the sinoatrial node, the atrioventricular node, and then those lower branches and Purkinje fibers. So this is just showing the signal being sent from the sinoatrial node to the atrioventricular node and then down towards the ventricles. It makes a little bit more sense if we overlay that concept with a more anatomically correct heart that's actually doing its job. So here we see that signal going from the sinoatrial node to the atrioventricular node through all those branches to the Purkinje fibers, and we have ventricular contraction. So you can see that as the signal is sent, as that depolarization event happens in the atria, we have atrial contraction, so atrial systole, and then atrial diastole, which is kind of happening around the same time as ventricular contraction. So the atrial relaxation, that repolarization, kind of gets masked when we look at this electrochemically by that ventricular contraction. But then we have the ventricular relaxation and the refilling of that chamber. So again, the goal of these signals being sent is to do something with the muscles that moves the blood. That kind of quantification and measurement process is what we're going to spend next Friday for my Friday class, and also next Monday doing. So kind of thinking more about these EKGs and how we measure them. So I wanted to take just a moment to remind you of sodium potassium pumps, as well as action potential, to kind of set you up for the movement of certain solutes, certain electrolytes, because it's a little bit different in cardiac action potentials than it is in neuronal action potentials. So remember that we have these sodium potassium pumps that are important for maintaining membrane potentials. They create a very strong concentration of sodium outside the cell, and then a strong concentration of potassium ion inside the cell. So they are actively pumping sodium ions out into the intercellular fluid. There's already a lot of them present, so that requires energy. We have a lot of potassium ions present inside of our cytosol, so moving even more potassium ions there requires energy. So we have this ATP being cleaved, we're releasing an inorganic phosphate. So to kind of verbalize that and have the words to what I was just saying, the concentration of sodium ion is higher outside the cell, the concentration of potassium ion is higher inside the cell. Both of those are moving against their concentration gradients. When you have these sodium potassium pumps, 3 sodium and 1 ATP are binding to the pump. That ATP is split and the sodium ions are moved outside. There's a conformational change. Potassium from outside the cell binds to that pump and then is transported inside the cell. So we have this ion balance that is key for maintaining that membrane potential. And as voltage-gated ion channels start to open, we then have sodium flooding into the cell and potassium flooding out along their concentration gradients. So when we think about that action potential, that is a change in voltage of a cell membrane in response to a stimulus that results in the transmission of an electrical signal. So sometimes we have those channels opening, different kind of movement of ions. Sometimes it doesn't quite reach threshold, but if it does reach threshold, then this signal is sent. And so we see this rapid depolarization and then repolarization of the membrane. The left image is going to be a GIF of a neuron, so you can kind of see that cycling of the charge going along the axon. So we have that depolarization and repolarization event. And if we track that by measuring it, we start to see this plot where we have depolarization going from that negative charge with a strong polarity, a strong concentration gradient, ions spilling across along their concentration gradient, and the membrane potential shooting way up to around 30 millivolts per neurons. And then it's going to actually become hyperpolarized as all those ions start to adjust. There's a reason why I'm getting into this. I know it seems like I just totally switched gears, but I really want you to be able to understand the ion flow in the cardiac conducting system and how it's a little bit different from what you've seen before. So we have this rapid hyperpolarization after the depolarization where we overshoot that resting membrane potential, and then things balance out again. So if you're at resting membrane potential, then we have a signal that is able to be sent. But if you are anywhere else, if you're being depolarized or repolarized or hyperpolarized or not quite back to resting membrane potential, you can't really send a signal. So we see that depolarization, repolarization, and hyperpolarization over time, and it does take time to get back to that resting membrane potential. So the time that's required before you can actually send a signal again, there's the absolute refractory period where we have that depolarization happening and then going into hyperpolarization. The entire kind of length of time from the end of that absolute refractory period until when we have all the ions back to where they're supposed to be gets into the relative refractory period. So we have these extended refractory periods where we can't send another signal until everything is back to the way it was. The potassium channels tend to lag behind a little bit. There's differences in the structure of those voltage-gated channels, so it takes a while for the potassium to get back to where it's supposed to be. It gets even more complicated in the heart because we're also worrying about these slow calcium channels. So those open up after the rapid depolarization, and they tend to stay open for quite some time. So not only do we have these slow poke potassium channels, we have these slow calcium channels, and we have this extended refractory period. We have a longer relative refractory period and a huge absolute refractory period. So overall, the entire refractory period takes a long time. It's extended. The purpose of that, or the thing that's accomplished through that, is that those myocardial contractile cells have a lot of time to fully contract. We get to avoid premature contractions, which are called PVCs. You can actually recognize those in EKGs, and we'll practice looking at them over the coming weeks. So we have this full contraction before we have another electrochemical event. So before contraction is triggered again, we make sure that we fully contract and fully relax that skeletal muscle. So now that you know a little bit more about those actual signals that are being sent, we're going to walk our way through the different parts of the cardiac conducting system. On an exam, you should be able to identify the correct order of this flow through the cardiac conducting system, starting with the sinoatrial node and then finishing with ventricular contraction. So this sinoatrial node has the highest rate of depolarization of this cardiac conducting system. It's what we call the pacemaker of the heart. So that pacemaker term is probably familiar to you from a lot of different contexts, but think about it as making the pace, setting the pace. It sets our sinus rhythm, that normal electrical pattern that's followed by contraction of the heart. So when the system is at rest, we don't have a signal being sent just yet. We have that extended refractory period. So the SA node is just kind of chilling. Then it initiates an action potential that goes across the atria. So we see that happening in step 2. Into step 3, that signal is being sent to the atrioventricular or AV node. So SA node, then AV node, that's located between the atria right before we get to the ventricles, and it's basically going to be receiving those action potentials and passing them on to the AV bundle. So just like how those kind of depolarization and signaling events happen in the nervous system and continue along, happen postsynaptically, we have this movement and passing on this kind of hot potato of the action potentials in the cardiac conducting system. So that AV node is receiving that electrochemical impulse, that action potential from the SA node, and then it's passing it on to the atrioventricular or AV bundle. So when we have this movement from the SA node to the AV node, there's a slight delay that allows the atria to completely pump blood. And if we think back to that GIF that we saw when I first overlaid the cardiac conducting system with the actively pumping heart, there was the signal and then a pause and that full contraction of the atria before we start sending the signal on, because you want to make sure you get as much blood as possible coming in from the atria before you pump that out to the from the ventricles to the rest of the body. So once we get into the atrioventricular bundle, it's just another chain of hot potato with the signal. So the AV bundle you might also see referred to as the bundle of His or the His bundle. So that is then going to receive that signal, pass it on to the bundle branches, which then passes it on to the Purkinje fibers. So the impulse transmitted to the AV bundle, the bundle branches and the Purkinje fibers, and finally the Purkinje fibers are responsible for sending that impulse to the contractile fibers of the ventricle. So getting back to that myocardial contractile cells and actually receiving that signal in the ventricles. So at that point, we have ventricular contraction. So you should know that order of SE node, AV node, AV bundle, bundle branches, Purkinje fibers, ventricular contraction. So when we start to think about how we look at this clinically, we're going to be thinking about measuring those depolarization and repolarization events, making sure that the timing of them, those pauses to allow for full contraction, are correct. Next week, we're going to be talking a lot about EKGs. So throughout this lecture, I've been saying EKG a lot, but you oftentimes, especially in the United States, hear it referred to as an ECG. EKG is from German, ECG is from American English, so they both mean the same thing, electrocardiogram. They just have slightly different spellings, but it's a way of monitoring that cardiac conducting cycle and tying it together with the cardiac cycle. So in lab especially, you're going to be working a lot with EKGs. You're going to be using something called the 8-step method, which we'll walk you through. So on the lab exams, you're going to have a lot of supporting information in front of you. You'll have to kind of interpret those EKGs. In lecture, it's going to be more theoretical information. I'm not going to give you an EKG and have you interpret it. So the just like general questions relating EKGs to the cardiac cycle apply for lecture and lab. The actual interpretation of it will take place in lab. So you can kind of start to see, I'll revisit this slide in the next lecture. When we say that term segment, those are regions between different waves. Intervals are a segment and at least one wave. And we have those different waves, those different patterns emerging at different parts of the cardiac cycle. So over the next few lectures, we'll really, and labs, we'll really make sure that we understand these EKGs and ground them together with the cardiac cycle. Lecture was a lot longer than I anticipated. Sorry about that. It does, though, set you up for a lot of physiology, so I'm hopeful that you watched until the end. Because it is important to really have that solid basis in anatomy to get into physiology. So please make sure you complete the online reflection. Monday class that's due on Monday, Friday class that's due on Friday. Complete that online reflection looking back at your experience on the lecture exam and preparing for the lab exam. Make sure that you listen to this lecture and keep listening to it. Don't just listen to it once and assume that you have it down. You have the opportunity to go back and review this. Make sure you take advantage of that. I'd also recommend scanning through sections 19.2 to 19.4 to prepare for understanding electrocardiograms and the cardiac cycle. And as always, please email me or come to my office hours if you have any questions at all, or even if you don't have questions, if you're just confused, I'm here to support you.